this video we will discuss about a paper that talks about matrix stiffness of pancreatic cancer. So the goal of the paper is to find out how this matrix stiffness affects the pancreatic cancer treatment. For example, it affects mainly in two different ways. That is, it provides the drug resistance, and also it helps the tumor cells in immune evasion. So these two aspects that they have targeted, and they finally come to a point that by targeting this matrix stiffness, one can improve the drug delivery to the pancreatic cancers or pancreatic tumors. At the same time, they will reduce the ability of this immune evasion by the tumor cells. However, in this figure we will discuss how they discuss the characterizations of the stiffness of the pancreatic cancer and in the stroma and also in the adjacent tissues. So they take the data that they have presented is from the 3 different patients who are affected with pancreatic cancers. So in the panel A, we can see that the stiffness of the adjacent normal tissues is around 2.5 kilopascals, whereas for the case of pancreatic ductal adenocarcinoma, it is around more than 3 times higher on average. That is 7.5 kilopascal. So this indicates that the stiffness of the pancreatic tumors or pancreatic ductal adenocarcinoma is higher, and it's almost like 3 times higher compared to the adjacent tissues. Thereafter, they validated the measurements using this green staining dye and also this hematoxylin and eosin analysis of that point from where their stiffness is measured. And we can see that for the 12 kilopascals, it is like more tumor related features, whereas for the case of 3.54 and 6.85, they see more pink dye or indication of the stroma kind of features. And these are from this pancreatic tissues taken from the patient. Thereafter, they also checked using the masson staining about the collagen fibers in the adjacent tissues and also in the pancreatic cancer tissues, and then compare them with the more commonly used model for the pancreatic cancers, that is KPC model. And we can see that the collagen features that they have seen in the PDAC tissues, which is indicated by the blue, is similar to what they have seen in the KPC model, indicating that the KPC model replicates the TME or the tumor microenvironment features of this pancreatic ductal adenocarcinoma. So this is how they showed. And the supplemental figures, they have also shown the H&E staining of these adjacent tissues, and here is the representations of this green dye that they have used. So green dye will able to once they measure the stiffness at a particular point, they are able to map those points through H&E staining to find out where this stiffness measurement belongs to, whether it belongs to the pancreatic tumors or the stroma region or the adjacent tissues. So this is why this green dye is generally used. And in this panel C with adjacent tissues, we can see that the collagen signals or the blue signals are very low, indicating that here the stroma formation is not there, and also collagen is not found in abundant amount in the normal tissues, because more collagens means there is increase in the probability of increasing the stiffness of the tissues. So this could also correlate with the low stiffness that they have seen in the adjacent normal tissues, and also the stiffness increased, and one of the contributing factor for that could be collagen that we have discussed in our previous review paper. And before that, we have discussed one detailed paper also about how this cascade effect between collagen matrix stiffness and tumor interactions occurs. So I hope you understood the matrix characterizations of these pancreatic cancers stiffness. And if you need to understand more about their experiments in detail and wants to refer to the supplemental figures, you can go through the descriptions. You can go through the link that has been provided in the description. Thank you for your time.